I would like to present to you Natalie and Sruja, and they're going to be presenting on their research on the differentiation of pyramidal neurons in the prefrontal cortex. So welcome, Natalie and Sruja.
So I'm Sruja.
And I'm Natalie.
And today we're going to be talking about differentiating pyramidal neurons in the prefrontal cortex of the brain.
Okay, so like Sruja mentioned, probably the largest part of our project is the prefrontal cortex. That's the part of your brain that's up here, and it's responsible for all the executive functions that happen in your brain. So these are things like language processing and impulse control. It's what keeps you from crying in public, basically. But the most important part that we'd like you to remember is that it's responsible for the working memory.
Working memory in itself is a system that's responsible for information processing when you're performing a task. For example, if I'm remembering a phone number just after someone's told it to me and I'm writing it down, I'm using working memory in that situation. Working memory, therefore, is a very, very important part of our lives because we use it almost constantly, and therefore dysfunctional working memory is often symptomatic with psychiatric disorders such as PTSD, bipolar disorder, attention deficit, and schizophrenia.
So on the cellular level, the prefrontal cortex is made up of 6 layers, and these are made up of interneurons and pyramidal neurons. We don't really care about the interneurons right now. We're going to concentrate on the pyramidal neurons. And these are the ones that are shaped like pyramids, henceforth pyramidal neurons. And we like layer 5 because this is where the biggest pyramidal neurons are, and they're the primary communicators for all of the prefrontal cortex. They tell the rest of the brain what the prefrontal cortex wants. And so there's 2 types of them, type 1 and type 2, which Sruja will go into.
Right. So type 1 pyramidal neurons and type 2 pyramidal neurons can be differentiated based on their morphology, pedology, and electrophysiology. That sounds like a bunch of fancy words, but it basically means the anatomy of the cell, where the cell ends up sending its firing patterns to, and the last thing is how it does that. So first of all, type 1 pyramidal cells tend to have really thick tufted apical dendrites, and type 2 pyramidal neurons have very slender dendrites with little to no tufts at all. The next part is that the pedology part is that type 1 pyramidal neurons tend to go as far as the back brain stem, and it can communicate with the spinal cord or the basal pons. Conversely, the type 2 pyramidal neurons only stay within their region of the prefrontal cortex, and they communicate contralaterally, meaning to the other hemisphere. And lastly, type 1 pyramidal neurons have really excessive, exciting bursts of firing patterns that conversely type 2 pyramidal neurons have very rhythmic and consistent firing patterns.
Okay, so before we got, there's one more way to differentiate between the neurons, and that's through genetics. And so before we actually did our research, people in the Thomas lab that we're working in, they did a genomic study on the different types of neurons, and they extracted mRNA from the type 1 and the type 2 pyramidal neurons, and then they sequenced that and compared it with this database called DCON, which is a list of all the genes in the type 1 or type 2 pyramidal neurons that are highly expressed. And from that, we got some antibodies for our experiment that we were going to use to identify the different types of neurons. And so for the type 1 pyramidal neurons that Sruja was talking about, we use SMI32. And then for the type 2 pyramidal neurons, we use INHBA.
Right. So you're probably wondering how we came to that conclusion. Well, the thing is, these antibodies were determined based on the genetic compositions of the 2 cells. And even though they're both pyramidal neurons in the same area of the brain, they don't necessarily share the same functions necessarily. INHBA, which is used for type 2 pyramidal neurons, is a gene that codes for glycoproteins in the brain, and it's involved in tumor suppressor activity and the function of pituitary hormones in the brain. And it's manifested in this glycoprotein right here. So, and the other antibody that we're using is SMI32, and this one is used for the type 1 pyramidal neurons. If you remember what I told you about the morphology of the type 1 pyramidal neurons, you'll realize that they have very, very thick tufts of dendrites, and they're much longer and more prevalent in the anatomy of the cell. Therefore, they're going to have more neurofilaments. These neurofilaments will be highlighted when the antibody attaches to that cell. So the big picture here is that when we use these antibodies on the pyramidal neurons and we look at them under the lens, the INHBA antibody is going to highlight the glycoproteins, and SMI32 will be highlighting intermediate filaments, which is basically just the cytoskeleton.
So why do we care about all this? It's mostly because when we think about the brain, there's a lot of things that can go wrong with it, and some of these things are psychiatric disorders. And a lot of psychiatric disorders can occur because your working memory doesn't work. And since we were talking about these neurons, these neurons are the working memory. How they interact creates the working memory. So if we are able to understand the differences between these neurons more closely, then maybe we can better understand how psychiatric disorders work and maybe come up with better treatments for them.
Right. So the experimental goal of our study is to visually differentiate the type 1 and type 2 pyramidal neurons in a mouse study, and we're going to be doing that through the process of IHC. IHC, better known as immunohistochemistry, is a fancy way of saying, is a fancy way of using antibodies and fluorescent tags to light up different cells so that we can distinguish between them physiologically. And there are 3 steps to IHC. One, we're going to be using a blocking agent just to make sure that there's no non-specific binding. Then we'll be introducing a primary antibody and a secondary antibody. The secondary antibody will have a fluorescent tag that will allow us to see the neurons under the microscope.
So how these antibodies work, like she kind of explained it a little bit, that we're looking for a specific protein that we mentioned before. And so that protein is expressed, and then we get the antibody to tag to that protein, and that'll tell us where it is. But we can't necessarily see the antibody because it's really small. So then we attach another antibody to the first antibody, and that one has a fluorescent tag on it. That way we're able to see it under a microscope. So with our experiment, the first part of the experiment was that we had to get brain tissue for our experiment and prepare it for the IHC. And that is the adding of the antibodies, which is the main part. And then there's microscopy, which is just looking at the results of the IHC.
Right. So the first part of our procedure, slice preparation, involves brain removal. Although we didn't actually remove the brains because we're not qualified for that, we did have some of the grad students in the Thomas lab do transcardial perfusion for us. And how that works is basically we're taking standard lab mice that were grown in the animal breeding lab at UNC, and we're going to anesthetize them with carbon dioxide. And after that, we'll be injecting a saline solution into their heart. And what the saline solution does, it removes blood from the entire circulatory system. Blood is really important to make sure, that blood doesn't stain the brain when we're doing IHC because the primary antibodies can actually have a high affinity to it. So we don't want to be lighting up blood vessels in the brain, we want to be looking at neurons. So it's really important that we get all the blood out of the brains. After the saline solution has finished running full circuit, we'll be extracting the brains and we put them in a 4% paraformaldehyde solution, and this just fixes the brain and makes the tissue stronger for IHC. And later they're frozen overnight in isopentane, and they're frozen for at least 24 hours. The next day we'll be taking the brains out of the freezer, and we'll be taking them to basically an instrument called a cryostat. It cuts the brains into 30 micron thick slices, and these slices are immediately placed into well trays. And these well trays are lined with PT. PT is just a combination of 2 different liquids, phosphate buffered solution and Triton, and this will just keep the tissues clean and free of debris.
Okay, so I can talk about the fun part.
Secondary antibody. The secondary antibody has a fluorescent tag, like I mentioned before, and this is what we'll be using to see the neurons under the microscope with the help of a different wavelength of light. After we've introduced the secondary antibody, we'll be incubating it with aluminum foil, and this will make sure that it doesn't quench the fluorophores. The fluorophores are basically the fluorescent tags on the secondary antibodies. And then we'll be doing one more PT wash, and then we'll be introducing something called a hook stain. The hope stain basically lights up the cell nuclei in a blue color under UV light. It's not really necessary for what we're studying, but the reason we're using it is so that we can compare it to neurons under the microscope. When we're doing microscope work, if we find what looks to be like a potential pyramidal neuron, to identify it as a neuron and verify that it actually is a neuron and not just some random tissue, we can switch out the light and look at it under UV light. This will make sure that we're correctly identifying what we're seeing under the microscope. And then we'll do one more PBS rinse just to keep things clean, and then we get to start on microscope work.
So this is the really cool part. We got to see things like this. This is the pyramidal neuron that we're talking about, but we actually got to see it, which is nice. This is another one under red light, but we didn't get used to that in the results. So the first thing we do is when we go to the microscope, we check the background fluorescence. And when we look at it, we'll see if it glows really green. If it's like totally green and everything's green, then we want to rinse out some of that green. So we'll do more washes with the PT and do that. But if it's black and we can't see anything and there's no slice there that we can see, we have to add more secondary antibodies and hope that it will attach to the protein that we are looking for. And then we'll actually look for results. And this involves like looking on the layer 5, at the layer 5 pyramidal neurons and seeing if there was any specific binding and not just background. And then afterwards, we would mount the slides that way people could use them for future research.
So the results for the INHBA antibody, and this one was for the type 2 pyramidal neurons, we actually concluded that the INHBA antibody was rather ineffective and weak at identifying these neurons. And as you can see, the control slide and the experimental slide look rather similar in the amount of fluorescence there is. There's little to none specific binding, and there's just a bunch of background fluorescence, which means that the antibody didn't really function, and it didn't really share an affinity for the pyramidal neurons, so nothing could be seen under these antibodies.
So then the SMI32 was really cool. It showed a bunch of specific binding, especially down here you see all these dots. Those are actually neurons, and this is layer 5 that we were looking for. So that was really promising. And the even better part was that our control basically showed no fluorescence. I know up here it looks really black, but really there was a little bit of green, but you can't really see it. So that was really good that there was no background fluorescence, and it was all specific.
So what does it mean? Well, first of all, because the SMI32 antibody was so successful, we can determine from the results that it shares a really high attraction and affinity for the type 1 pyramidal neuron. This probably means that the intermediate filaments found in those cells are highly expressed, which also means that the SMI32 gene itself is a very important part of the type 1 pyramidal neuron. Conversely, the INHBA antibody, which didn't really highlight any pyramidal neurons in the microscope slides, we can determine that that has low affinity for type 1 pyramidal neurons, and that the glycoproteins that it was supposed to highlight didn't really happen. So there may be a chance that the glycoproteins aren't really highly expressed in type 2 pyramidal neurons. However, before we take this into account for future studies, we have to remember one thing. The INHBA antibody was tested at one specific concentration, 1 to 250 microliters. That was the ratio. And because this antibody is one of the first to be used in these kinds of studies, we can actually take a look at different concentrations and see if a higher or a lower ratio might better identify the type 2 pyramidal neurons.
So when we think about what could have affected the results, we immediately go to INHBA because it didn't work the way that we wanted it to. So the first thing was, this was the first time a group of researchers had used INHBA for this purpose. It had been used in other tissues before, mostly like with cancers and stuff, but it hadn't been like with pyramidal cells. And so that could have affected what concentration we picked, because we had to go off of other concentrations of other antibodies, and it wasn't specific for the INHBA. And then also the manufacturer, they recommended that we perform a heat-mediated antigen retrieval, which is fancy to say we should have prepared the slides with some heat and this buffer in order to make the antigens come out more. But we didn't do that because we didn't have enough time. And then also probably the most important thing that affected our results was that we were only able to do this once. If we had more time, we probably would have repeated it, and then we would have gotten a higher percentage of significant results.
Right. For future research, well, the first thing is because the FMI32 antibody was so successful in staining the type 1 pyramidal neurons, we can use that as a basis to go off and test other genes and other antibodies for identifying these cells. And remember, the big idea is to understand psychiatric disorders more. And because these cells are so involved in working memory, we're trying to understand their genetic composition, their physiology. So studying these antibodies and what kinds of genes actually make up the cell and their functions is really valuable. Another thing, like I mentioned before, because the INHBA antibody, we were the first ones to ever use it on pyramidal neurons, so we can actually test it in other concentrations to see if it actually might be successful in identifying type 2 pyramidal neurons. And the last thing, the decon gene database, where we actually got these antibodies from. The database actually has hundreds upon thousands of more genes that could be potentially expressed in pyramidal neurons. So there's so much research to be done, and we can't just stop here because understanding psychiatric disorders may be a very challenging task, but little by little, by understanding each little gene, we can eventually determine its function and therefore how working memory relates to psychiatric disorders. These are our references, our image citations. And first of all, we'd like to thank Dr. Mark Thomas. He's not here today, unfortunately, but he really gave us an amazing opportunity to study neuroscience under psychiatric disorders, because it's a really underdeveloped field, and we were lucky to have so many resources given to us to do research on this topic.
We would also like to thank everybody who works in his laboratory as well, because they were very good with guiding us through this whole process.
And next, we want to say thank you to our lovely student mentor, Emily Richard, because she helped us through all of the experiments, and she really guided us in the process and really taught us what immunohistochemistry actually looks like.
We'd both like to thank the Union Pacific Foundation for funding our research. They were both of our sponsors.
And I'd like to thank Wells Ranch for sponsoring my room and board here. I wouldn't be here at FSI without their help.
As well as the Adolf College Foundation, they sponsored my scholarship here, so I'm really grateful for that.
And we want to thank UNC for letting us use the facilities and equipment for our research.
And then also Lori Ball for putting this whole program together and making sure it stayed together.
And of course we want to thank Nicole for all the help she's given to us as an advisor in writing our research papers.
And then thank you to Sophia and Christopher for being great RAs.
And next we want to thank the FSIers. You guys have always been supportive of it. I guess we've all been supportive of each other collectively in our research, and it's really nice to see a group of students coming together to study science. And of course we want to thank friends and family, because without them we wouldn't be encouraged to study our burning passions for science.
And really we'd like to thank the mice, because they did donate their brains to science, so that's pretty serious, so we'd really like to thank them.
Is there any questions for us?
Did you guys choose to go